Question number 58. Given below are some techniques used in modern biology. Number 1, in vitro fertilization. Number 2, cryopreservation. And number 3 is tissue culture. Which kind of similarity is carried by all these techniques mentioned above? See, in vitro fertilization is that fertilization is carrying out in the lab, that is outside the animal body or the plant body. Alright, it's in petri dishes and tissues and all that lab apparatus. And cryopreservation, and once this is fertilized and then it is put inside the animal or plant body, the way it is. Alright, and then cryopreservation is nothing but preserving the tissues for their life, so that the cell activity is arrested and they can be used later on. So this is usually done in liquid nitrogen. Okay, at deep freeze temperature like about minus 148 degrees Celsius and beyond. Then tissue culture is nothing but a growing of callus from the explant, and then slowly developing into an entire individual. Okay, so it is carried out both in animals as well as in plants. So basically these techniques are nothing but a sort of in vitro techniques, or what you say, which are carried out in the laboratory outside the natural environment. It is carried out artificially. So this was the background. Now let's see which of these options are having similarity. Okay, now option A, all are in situ conservation methods. No, not at all. All are ex situ conservation methods. C, all techniques require ultra modern equipments and highly skilled personnel. No. And then all these are methods of conservation of extinct organism. No, this one is highly irrelevant. Now see, ex situ is somewhere we can say it's close by in vitro conservation, that is outside the natural conditions. And now this term, if we extend to the wider scopes, then it can be applied on our ecosystem as well, wherein we say that it's nothing but the off-site conservation, that is the process of protecting an endangered species from picking them up from their natural habitat and putting into some wild or maybe in artificially created suitable situations for them. So over here what we are doing is that by these means, in vitro fertilization or cryopreservation or tissue culture, we are just going against the natural laws, and slowly we are trying to develop any of the species of desired concern, um, uh, by using these techniques. Alright, so option B is the most preferred one. With that, moving on to next question.